Hi, everyone. My name is Caio Seguin. I'm a postdoc at Indiana University. And today I'll tell you a little bit about this work on modeling propagation of stimuli through the human connectome, done with this amazing team of collaborators. I also have a poster that I'll be presenting virtually on June 8th. So if you're interested, come by. I'll be talking about brain network communication models. And they try to answer this key question of how are signals communicated between gray matter regions through white matter connectivity. Here we have a representation of the human structural connectome, where each node indicates a gray matter region, and a link between two nodes is a white matter tract. And we can see that this network only encodes information on direct anatomical connections between regions. So as a result, the structural connectivity matrix is typically sparse. So we can't, based on this matrix alone, we can't really infer anything about regions that are anatomically unconnected. And this is where the network communication models come in. So the idea is to try to understand if, for instance, you stimulate region I and you read out an effect in region J, what are the paths along white matter that facilitated the communication of this perturbation from I to J? So these models provide a putative conceptualization of network communication with the goal of understanding that. And they really tend to be a simple strategy to find paths or sets of paths between nodes in a network. And they also deliberately abstract away biophysical details in order to try to gain some simplicity of explanation. And what they afford us is this, what I call the communication matrix, which now tells us for each pair of nodes, their ease of communication under a certain putative communication model. So the idea is that this matrix is dense, whereas the structural connectivity matrix is sparse, because now we can model communication between any two pairs of nodes. And there have been lots of different conceptualizations of network communication proposed in the network neuroscience literature. And they roughly fall in the spectrum from decentralized or diffusive measures to routing measures. The diffusive measures propose that communication is a sort of diffusive ebb and flow through the network, whereas the routing measures propose more of a message passing along efficient paths. And conceptually, all the models along this spectrum have pros and cons, and we don't really know which conceptualization of network communication best approximates biological neural signaling. So how do we move forward? How do we determine this? How do we make progress in finding out which of these models are more useful? So traditionally, model validation has been done trying to understand data from fMRI and both signals. But the problem here is that this data only encodes really slow activity fluctuations. So I set out to try to understand, can network communication models explain stimulus propagation at millisecond scale? So the scales inherent to brain communication, to brain computation. And to do this, I'm going to use intracranial EEG recordings from the FTRACT project. So I'm going to give you a crash course on the FTRACT project. So it was done on drug-resistant epilepsy patients that were being prepared for resective surgery. And the idea is that these patients are implanted with intracranial or SEEG electrodes to determine the epicenter of their epileptic seizures. And the SEEG electrodes can both record activity and deliver electrical stimulation. So the FTRACT project gathered data for more than 1,000 patients being prepared for surgery. And the key thing is that the number and the position of the electrodes varied for each patient. And this is really the strength of this project, because by combining this data across all these different patients, the FTRACT team created this group level whole brain matrices of stimulus response measures. So this is a schematic of how the FTRACT project generally works. The idea is if you deliver a stimulation in this region and you read out the effects of that stimulation in two regions, we can even have a significant response, which is a response above a certain Z-scored cortical evokes potential, or you can have a non-significant response. So this, based on this really simple setup, by combining the data across all participants, we can create this response probability matrix, which tells us if I stimulate region I, what is the probability that I'll observe a response in region J? And this is what I'm going to try to be modeling in this talk. Okay, so the key idea is to, so we've talked about the response probability matrix from abstract. And what we did is we used human connectome project data to map structural connectome. So we derived structural connectivity weights from tertography. And we also derived functional connectivity from fMRI. And we derived the interregional Euclidean distances between parcels in our parcellation. And essentially, the key idea is to use the information in panel B to model the information in panel A, right? So can we compute measures on a normative structural connectome to try to explain stimulus propagation in the abstract data? So the really cool thing about the abstract data is that by combining this data across all of these patients, you really get response probability estimates that cover the entire cortical surface. So here I'm showing each entry here is a pair of regions for which we have at least 100 stimulation experiments, for which we can have a reliable estimate of probability. And here, if we sum along the rows and the columns of this matrix, we get a distribution of stimulation sites and a distribution of recording sites. And although that distribution is spatially heterogeneous, because they fall within common epileptic centers in the brain, they still allow us to do a whole brain mapping of stimulus propagation, which is really cool. So what we did is we divided pairs of regions into regions that are likely to be anatomically connected and likely to be anatomically unconnected based on our human connectome project sample. And we can see that for regions that are anatomically connected, the probability has we have a higher mean of probabilities, statistically higher, which kind of makes sense of activation. And then we correlated these with these simple measures of Euclidean distance in green, functional connectivity in pink, and structural connectivity in red. So this is done for all pairs of regions. This is done for anatomically connected, and this is done for anatomically unconnected regions. And what we can see is that in all cases, the Euclidean distances between regions seems to have a really strong influence. So here, sorry, on the vertical axis, we have correlation to P, or the absolute value of the correlation to P. So the distances between stimulation and recording sites seems to be really important. Oops, sorry. And functional and structural connectivity provide explanatory power as well, but not to the same extent. So now we're bringing in the network communication models. So we chose 5 putative conceptualizations ranging from routing to diffusion. And all of these were computed on the structural connectome, right? So we want to understand how the white matter connectivity constrains stimulus propagation. So the model spanning routing to diffusion. And because Euclidean distance had such strong performance, we considered both full and partial correlations, the latter controlling for the effect of Euclidean distance. And what we found is that Euclidean distance remains the top predictor in all cases. But when we included network communication models, we observed really high partial correlations, factoring out the effect of distance. And that is to say that the white matter architecture and the dynamics that are engendered by these network communication models are explaining variance in response probability above and beyond the effect of distance. And another key thing is that if you just compare the performance of structural connectivity, which is below here, to that of network communication models, we can see that by considering network communication, we really improve on what structural connectivity can tell us. Especially in the case of anatomically unconnected regions, structural connectivity has nothing to tell us. And the communication measures afford us some predictive utility. So yes, already said that. And search information really was the top model. And I'm going to get to the mechanics of search information in a bit, which is the blue one here. So what would happen if we put all of these measures of network, structural, functional connectivity, Euclidean distance, And we let them fight to explain the variance, because so far we've only done univariate statistical tests, right? So the goal here is to try to find a sparse set of predictors that can predict response probability. So to do that, we use the multivariate machine learning method that penalizes model complexity, is a loss-through regression. And what we found here is that, so this is out of sample prediction accuracy, and this is shown in a scatter plot. So we can get really good prediction accuracy. We're explaining about half of the variance, a little bit more than half of the variance in probability, with a really sparse set of predictors. So all the predictors were included here, but only 3 came out in the end. So it was a parsimonious combination of structural. So this is communication using the search information measure based on structural connectivity, geometric, Euclidean distance, and functional factors. So using only these 3 things, we could predict response probability really well. Search information was again the best model. But what is search information anyway, right? So in technical terms, it's related to the probability that a random walk will travel between 2 regions via the shortest path. But in the context of brain stimulation, it can be understood as the propensity of signal dispersion on the path from the stimulation site to the recording site. So the idea is if we have 3 regions here, A, B, and C, and we stimulate these 3 regions, and we want to measure the response in the target region T. You can see that the path from all of these regions to T has the same length, but the path along starting from A has nodes that are a lot more connected to the rest of the network. And that engenders more opportunities for the perturbation delivered in A to be diluted as it moves along. So here we have the search information from A to T is the highest, and the search information from C to T is the lowest because the nodes along this path have very low degree. So the idea is that stimulating C will lead to a higher probability of activation in T, which I think is a cool insight. So to wrap it up, we provide empirical evidence that network communication models infer from structural connectivity based on MRI, can explain variance in stimulus propagation measured at millisecond scales. Spatial proximity between recording and target sites was really important. FC provided important information, and I think that this is capturing aspects of the underlying dynamics that are happening when you deliver that perturbation. And search information was the best conceptualization of network communication. And this hints that perhaps certain network communication models might be more useful than others. And again, check out my poster on June 8th if you want to come and talk to me more about it. All right, thank you.